So in this talk, I'm going to walk you through, in fact, give you an overview of what's involved in germline variant discovery. How these talks are organized is day one, we're just doing introductions to all of these pipelines. Don't worry if this is too much information. We're going to go over each tool and each pipeline again each day. So day two is going to be all germline focused, and we'll talk more in detail about each tool and go over this set of slides again tomorrow. And then day three, we'll focus on somatic. So if it's too much information, don't worry. There's more to come. We'll drive home the information. All right. As Andre mentioned, there are these major variant classes and various tools for each, for germline and somatic pipelines, and for short variants, there's a different tool, and for copy number and structural variant, and so on and so forth. For germline, in this workshop, we are going to focus on Haplotype Collar. So backing up a little bit, what's the difference between germline and somatic? Germline is essentially all the variants that you are born with, that you inherit from your parents, one half from your mother and the other half from your father. There are also some germline variants that are unique to each person, and those are only in the range of about 30. But other than that, it's all that you inherit. And so for this workshop, we are going to focus on short variants, which are point mutations and insertions and deletions less than 50 base pairs. And this is an arbitrary threshold that we've set for short variant discovery, just 50 base pairs. So this is the overall best practices pipeline for germline variant discovery. We went over this first column, which is data pre-processing. We spoke about that. In this talk, I'm going to give you more of an overview of the variant calling, which is the second column, and the third column is filtering your variants and refining the genotypes that are called. So the key player in germline variant discovery is this tool, Haplotype Collar. Under the hood, Haplotype Collar essentially follows these 4 steps. The first step is identifying active regions. What Haplotype Collar essentially is doing is looking at your genome and saying, I'm going to focus on the regions that have most variation. That's because a lot of your genome is very similar to your reference. So you want to concentrate on only the regions that have variation to make it more efficient. And that's essentially what it's doing in active regions, identifying active regions. And then it does local realignment using a graph-based assembly to create haplotypes from reads around complex sequencing regions in the genome. And then it takes each of these reads and then determines likelihoods for each reads against the haplotypes that it creates. We will go into more details of this tomorrow. We're going to have one talk focusing only on Haplotype Collar, and Geraldine will talk to you more about how all of this works. But essentially, the fourth step is getting the genotype for your calls. Andre went over joint calling a little bit. So why do we do it? We do this because a single genome in itself is not giving you much information. For example, you find a variant in a sample, and you want to see if it's associated with any disease. How do you know that a variant call has some biological consequence? Maybe that variant is present in one of the parent, and they're healthy and they don't show that the disease phenotype. Or maybe in that population, most people have that variant and they don't have the disease phenotype. So adding that family and population data will help you filter out all of these variants. And so that's essentially the idea behind doing joint analysis. And you want to focus on variants that are rare in a population because those might have some something to do with any disease-causing phenotype. So back in the day, how we did haplotype collar and join calling together. So if you say had 10 samples, haplotype collar would create a graph for all the 10 samples and then do join calling for the entire data set. And so if you increase the number of samples, the graph just got bigger and bigger. So this made haplotype collar very time consuming. And also, if you say had one way of sequencing, which gave you 10 samples, and then you got the second way of sequencing and needed to add samples to your original data, you needed to go through the haplotype collar process all over again. And this just made it very time consuming. And so we came up with this two-step process. In this two-step process, we essentially run haplotype collar on each sample just once and create a GVCF file. I'll talk more about what a GVCF file is, but essentially the idea here was to, in case you wanted to add more samples to your data, you would need to run haplotype collar just once and create this GVCF file, and then you combine all the GVCF files and do joint genotyping on that entire set. So in the GVCF mode, it's the same 4 steps: identifying active regions, creating haplotypes, determining the likelihoods of each read against these haplotypes, and then genotyping. And now to talk a little more about the GVCF file that haplotype collar creates. GVCF file is essentially a genomics VCF file. It follows the same format specification as a VCF file, except that in a GVCF, it has information of all the positions in your genome. Now you might ask, why is that? Why do we need, for example, here, this is a GVCF, and it has information about the reference. And the yellow parts are all variants, but the blue ones are information about the reference. So the question here is, why do we need that information? So think about it. If you had multiple samples in your data set, and you had one variant, and you compare that variant to other samples, how do you know? Okay, so you have multiple samples, and one of the samples has a variant. Now, that variant becomes interesting if every other sample in your data set or in your cohort has good, high-quality reference information. So now you know that in every sample, that region was well covered, has good depth, but in one sample, that variant is seen, it's not seen in the other samples. So that variant gets interesting. Or in cases where you don't see any variant, how can you be sure that there's no variant in that position because there's actually no variant, or because sequencing didn't cover that region? So having information about each genomic location in your genome helps clarify those questions. All right, so now you have a GVCF, you've done joint calling on it, but that's not the end of the road. You also need to filter your variants and also refine your genotypes. And we'll talk more about that. So there are different ways you can filter your variants. In this slide, what we're showing is essentially using a tool called VQSR, which takes 7 different annotations and uses a combination of those to see what are good variants and what passes those variants. And all these pink dots are the ones that have passed the threshold for being good variance. And so if you wanted to do this with, say, 2 annotations, you take any 2 random annotations on the X and Y axis, and if you try to form a box to enclose the good variance, there's no real good way to do that with 2 annotations. So if you try to like adjust these 2 lines in such a way that you want to encapsulate the good past variance, it gets a little difficult. And the idea here is that, and what we're trying to get at, is that with hard filtering, if you had to manually decide the thresholds for various annotations, it gets hard to, you will miss out on some good variance. And so we have these machine learning tools that will help you do variant filtering, and we'll go into more details about that tomorrow. All right, so we spoke about this earlier, adding population priors or family priors helps you. How do we get rid of that? Okay, never mind. So adding, is that something I did? No. Yeah, this works. All right. So yeah, you can add population priors and family priors, and we spoke about this to make your calls better, to refine the genotype. And so this is an example where we found 417 de novo mutations. And just by looking at the number, you know that that doesn't make sense. Like I said, in each person, there's about 30 de novo mutations that are unique to just you. But 417, that seems like a very large number. So we applied population priors and family priors to this data set, and that number came down to 17. And then when we used just high confidence de novo mutations, then we got a good a number of 8 de novo mutations. Now that's more believable. That's not working. Yeah. All right. Andre went over this already. We have a tool called Funcutator, which applies functional annotation to your variants, essentially giving you information about what are the total SNPs in your reads, what is the ratio of non-synonymous to synonymous variants. In this, it's obvious that most of the genome is in the intronic region, so most of the variants that you see are also in the intronic, and then you have these various annotations that you can apply to your variants. And finally, once you have a call set, at the end you also want to evaluate your call set and see how good were my calls, how sensitive my calls were, how specific were they, what is the ratio of true positives to false positives or false negative. And to do this, you would need a true data set. Again, we will do concordance analysis. We will do some hands-on tutorials on it and see how you can figure out or evaluate your call set tomorrow. But essentially, the idea here is that you want to be as close to true positives and away from false positives, but also not losing out on any, basically getting too many false negative. So essentially, you want to be close to this steep curve, and how you get to that is using various filtering techniques. And that's an overview of what is comprised in the Germline variant discovery best practices pipeline. And that's it. If you have any questions, too much information, we will go through all this again tomorrow. So. Sorry, can you say that? Uh-huh.
When you say active area of research, do you mean what is this used for, or what are we working on to improve it? I would say that the biggest amount of development for germline short variants is probably around filtering. At this point, well, filtering and then other types of data. So let me talk about filtering first. Most of the variant calling proper is really well understood in terms of the types of error, types of problems we encounter. We're capable of making very sensitive call sets. The difficulty, for the most part, is in is the filtering parts. And now for filtering, we do a pretty good job when we have a lot of samples. And so for multi-sample analysis, for like, let's say a few dozen up to a few dozen thousand samples, we have that pretty well in hand. Where it gets complicated is at extremely high amount of data and very low amounts of data. So for the very low amount of data, I would say, well, actually, let me start with the higher end. When you have extremely large amounts of samples, like we're starting to get into the 100,000 whole genomes, it's a lot of data. There's a lot that we can parallelize, but when we get to the filtering stage, we have there's some technical challenges of scaling up. Some of it is just because the size of the output file, for example, if you're outputting a classic VCF file for a multi-sample cohort on 100,000 whole genomes, you're looking at terabytes and terabytes. You can't really make a single file for that. And so we're looking at options for like database formats, where instead of outputting a VCF, you're outputting a database of some sort. One of the options is to use, for example, because we're working on the cloud and we're working with Google Cloud infrastructure for the most part in our production needs, we're looking at things like Google BigQuery, which is a database format. One of the questions around that is, if we start using that kind of systematically, how do we still support a user community that's not on Google? And what are the equivalent services that we can connect to or that we can support? So that's kind of an area where there's some active thought and development ongoing. The other side of the range is more around very small amounts of data. When you have a single whole genome, the algorithms that we have traditionally used for filtering don't work as well. And so that's why we look to other types of machine learning, including deep learning. And so now we have a workflow that is geared specifically for single genome analysis, where the filtering part is using neural networks. So it's convolutional neural networks, and we'll have a presentation that touches on that tomorrow, where that performs much better if you have only a single sample's worth. So for now, multi-sample, we use the classic VQSR. Single sample, we use CNN. The nice thing is that the neural networks-based approach should also be applicable for areas that have traditionally been challenging for GATK tools, like non-model organisms, where it's often also a problem of insufficient amount of data or known variants. And so there are some perspectives for being able to use the neural networks approach to those use cases. So there's a fair amount that's happening there. And then I would say the connection to the tertiary analysis of what do you do with the variant calls. One of the active areas of development is around For example, if you want to do a genome-wide association study with the outputs of your variant calling pipeline, we're working with a team that's also at the Broad that develops a toolkit called HAIL. And HAIL is it's not really tools in a traditional sense, it's more a development toolkit that provides you with a kind of library of genetics analysis functionality that is optimized for using Spark infrastructure. So Spark is a system for massively parallelizing operations. And so the nice thing about HAIL is that it allows you to run genetics analysis at a much larger scale and much faster. So you can do kind of larger scale analysis in a much more scalable way. And so there's some work that happening around the connection. How do we provide a more seamless transition from getting the call set out of GATK and actually starting to use it? That's where a lot of development is going right now. Andre, does that sound like you're part of the development team? I'm not, so just want to get a gut check. That's about right.
Another things to mention that I know is currently under development is another person just joined GTK team who actually used to work on GK, Kieran. And he is working on adding some things to assembly graph to kind of have a better representation of the haplotypes. And specifically, he's trying to do that to be able to call variants on long read sequencing. So that's one of the things.
That is the other data types. Thank you so much for mentioning that. Because for a very long time, GATK has been very specifically developed for Illumina short read data. Although there was a poster like 5 years ago about adapting the pipeline for PacBio. A lot of water under the bridge since then. We didn't do anything with PacBio data for years, and now Kieran, and it's interesting, Kieran was one of the original founders of GATK. I think he was one of the, might even have been a first author of one of the papers. I forget. Anyway, he was an original developer, went off and developed his career elsewhere, and then has come back and is now bringing long reads analysis to GATK, which is, I think, going to be really important. Sure.
So, how about RNA Seek?
Can you say that again? Oh, RNA Seek? Oh, yeah, there was some other questions around RNA Seek. So within the development team, I don't think we're actively working on it. However, we are part of a wider group within the Broad that's called the Data Sciences Platform. And the data engineering team that we work with is part of the Human Cell Atlas project. And so they're actually responsible for productionizing all the mostly single-cell RNA-seq analysis pipelines for the HTA project. And so we are working with them to figure out how to bring our pipelines together kind of in the same place. And the plan right now is to load all of the HCA pipelines in Terra and provide the same kind of example workspaces to make it easier for people to use those pipelines. Andre, did you want to add something about that?
Yeah, we do have a team actually in GDK who they do single cell RNA sequencing analysis. They mostly are responsible for pre-processing, and I guess they do jointly work with other people. Right? Yeah, Murtash is started.
Oh, he's doing that now. Okay.
Yeah.
I'm a little out of date. Sorry about that.
Yeah. But not bulk RNA, if that's what you said.
Yeah, there's a lot that's happening in the development group, and from month to month, sometimes I find myself slightly out of date. It's what, like 30 people at this point in the methods team? Yeah, I feel like it keeps growing, and I turn around and now they're working on new problems that they weren't before, which is cool, but it's a lot to keep track of. Sorry. Any other questions?
Regarding the joint calling,
Mhm.
in a typical big project, so there's also going just maybe a few thousands first and those, and during the evaluation, I think that protocols and sequencing machines may be changing. But if you were to just go with the GVCF approach, like you create one GVCF file from one sample, and then so is there some kind of limitation of adding the files that were not like sequenced in a similar way as the ones in the main? So are we running into danger of mixing together in the end?
Yeah, so the question is about like when you have a large project and you're accumulating samples over time and processing them over time, and maybe some things change. So what's the risk there? For us, we try to, and it kind of goes back to my earlier answer about the batch effects. We do try to process everything in the same way. And so when we're doing a joint calling, we actually pin, like all of those samples we process them in the same way. There are sometimes a few minor changes that we allow if there's been a small bug fix and we understand really well what has happened and what has changed. But for the most part, everything in a particular project gets processed in exactly the same way. In some cases, that means reprocessing and recalling some things that were done previously. But we want to do that to eliminate batch effects as much as possible. Even like the nomads call sets. So nomad it stands for genome aggregation database, and it's at this point it combines data from 100,000 exomes, and I think we're at 70,000 whole genomes within the latest version that has been called. The exomes and the whole genomes are called separately. We don't support joint calling for exomes and genomes together. In that case, those things are aggregated afterwards. But for exomes and whole genomes, for each of those separately, all of them are called in the same way, processed and called in the same way. Yeah. Yeah, so we use the same pipeline, and it does mean that sometimes by the time you're sequencing your 50,000th genome, the tools have evolved. We still use the older version, or we decide the change is big enough to reprocess everything with the latest. But it is true that the big advantage of GVCF is that you can do that, incremental processing. But we really don't recommend mixing versions within the same project, just because of the risk of batch effects.
You mentioned about a single sample you use the CNN approach. Is it different from the one that Google uses, like deep learning?
That's a great question. So Google came out, the Google team, Google Genomics or something, team came out with a variant collar called Deep Variance, I think 2 years ago, that is based on a deep learning approach. And it's interesting because that one was written by two people who used to be GATK developers, Mark DePristo and Ryan Poplin, who were part of the original GATK team, gone off to Google. And it's mostly it's a hybrid approach that uses, that works in 2 steps, where the first steps is basically Haplotype Collar, which makes sense because those were the guys who wrote Haplotype Collar in the first place. And then the second step is a deep learning based classification of the variants that happens. The CNN approach is kind of similar in the sense that we're also doing Haplotype Collar first. We're just doing it explicitly as a separate step, and then applying CNN. I'm not the right person to talk to the the comparison between how the algorithm is implemented, but it is similar concepts that are used. But our CNN works better. I have to say that. We have some benchmarking. It's honestly not a huge difference. You're getting into very small decimal points in terms of the difference that you get, because they're very similar.
And if you want any specific questions about CNN, you could post on the forum, and we'll have like the developer of CNN look into that and give you an answer directly. So yeah, use the forum if any of your questions aren't answered here.
In principle, it should be applicable as well. The thing is, we haven't done it yet, so we can't. We haven't validated how well it works and how, because you have kind of different error modes, different problems to deal with in the somatic context, and so you could conceive that the approach would be applicable, but there you might need to have some tweaks. We just haven't done that work yet. Yeah, so if we look, the biggest difference that you get is on single whole genome, single whole genome case, where VQSR doesn't do well, and there, yeah, it's still a few percentage points in terms of, because it's always that trade-off between sensitivity and specificity, and being able to really finely differentiate artifacts from real variance. And we just find that the deep learning approach performs better in those cases where you just don't have as much data to feed into VQSR. But these are often like fairly small differences. The thing is, it really depends a lot on your application. If you're doing large-scale population genetics, there's often not a big advantage in either case. But if you're looking at rare disease variants and you need very high kind of fidelity, that's where it has a bigger impact to be able to have that much higher sensitivity. Go ahead.
Multiple additional samples available in the laboratory. We have the single sample and leave it up with the others, or you apply the CNN algorithm.
I don't think I followed you. Can you repeat that one time?
If you have kind of identical samples available in the laboratory, so would it still be better to kind of pull everything together, or to run the CNN algorithm by individual samples?
Oh, I see. You're saying if you have samples, like normal samples from the same person? Oh yeah, yeah. Yeah, if you have additional samples that you can use, it's definitely worth. Yeah, it's a hard question. So there is a paper about haplotype collar that authored by Laura Gauthier and others on the team, but she did titration experiments basically to see, as you add more samples, what's your increase in the fidelity of the variant calling, and what, if any, drawbacks might start to creep in. As I recall, but she didn't compare that to the deep learning approach, because CNN wasn't ready at that time. So it's more it tracks kind of the gains. I would say you start seeing benefits very early on of having multiple samples, especially with a few dozen or a few hundred samples. That's where you see the biggest, fastest increase of the amount of samples that you're looking at in your cohort, and then it starts to tail off. And once you're above a few hundreds, the gains get smaller and smaller, because that's where it's really on the rarer variants that you start seeing an effect. Now, if you're looking for very rare things, you need to get into the tens of thousands of samples to have a chance of seeing them again in another person. But the bulk of the gains are around a few hundred samples. Now, if you're talking about like a handful of samples, it's likely that the CNN approach might actually already do better and be more worth it for you from an implementation standpoint. Because also when you're looking in a clinical context, there's a reluctance to use other unrelated samples, because then when you, at least depending on the type of clinical pipeline you have, in some cases you can't use other unrelated samples, because if you redo your analysis with other background samples, the results are not exactly reproducible. There's a little bit of variation that's introduced by what you're using as a reference. Now, if you're always using the same exact unrelated samples, that's fine, but you can't just add more as you get more coming into your facility, because that changes the results for a patient. That gets into more the regulatory side of things. It depends on what the approval process looks like for your pipeline.
And currently, CNN can be used on only one sample, but the future CNN is going to be to use it on multiple samples.
But I think you can use it on more than one. It's just, the advantage is demonstrated on a single one, I think, isn't it? Am I wrong?
So
hang on a tiny bit.
So I know that Sam's tested it on genome, whole genome, exome, Somatic, and Germline, and it's all worked really well. And that's what we're going to add to our best practices as well. But on more than single sample, that's something that's
but it is technically feasible. It's just that it's not validated as being the way to do it.
Right.
Yeah, that's what I thought. Sorry, you were saying? We're just checking something here. That's a good question. I'm not sure. I will find out. Right. I think it's reproducible internally. Do you have?
So tomorrow, this still works. Okay.
Yeah.
So tomorrow we're doing this tutorial on germline filtering, where we will use CNN to filter variants. And so we'll have this workspace set up where you can test different settings of CNN, tried multiple times on different data sets.
But I think the question is, if you run it on the exact same data set, does it spit out the exact same result every time? So is it deterministic? I believe it should be. Given, it depends if there's sometimes some of these algorithms use like random seeds and stuff like that, which introduce some variability, but there's usually an option to use a specific one to enforce determinism. I'll look into it. I'll get back to you tomorrow on the exact determinism of CNN. Usually we try to make those things as deterministic as possible. My comment on the variability was more, if you're using different reference samples, like different non-matched normals, then you will see some differences. Any more questions? Or can we do lunch? All right, let's do lunch.